This is the third episode of an exciting new series led by two of the Plant Genes Steering Committee members, Ken Sub Lee and Nigel Taylor. If you guys just want to give us a wave, people should be able to see you. This is not a traditional workshop, but it's more like Albert giving us a tour of his lab and his protocol. And this is an interactive session. We'll be stopping throughout the protocol to take audience questions. So with that, I'd like to introduce today's speaker, Dr. Albert Kausch. Dr. Kausch is a professor at the University of Rhode Island in the Department of Cell and Molecular Biology with a research focus on molecular improvement and gene discovery in plants. Dr. Kausch was an early pioneer in plant transformation with now 40 years of experience in plant transformation technology development for commercial and academic research, as well as agricultural biotechnology. His broad industry experience extends from working with large multinational seed companies in a vast variety of areas. He maintains several active research collaborations on plant transformation and genome editing technology development through his laboratory at the University of Rhode Island. Dr. Kausch's contributions include technology and events generated through genetic modification, traditional breeding, genomics, and gene editing of various crop plants that are now grown commercially worldwide. Today's protocol has 8 sections. We'll go through each section and then pause for questions. Questions will only be taken from the Q&A box. You should see that at the bottom of your Zoom screen. I'll be monitoring the chat and moderating questions for the sake of time. If we don't get to your question in this section, we'll try to get to it at the end with any leftover time that we have. And any information presented today will be available on the Plantgene website along with a recording. So with that, I'll share the slides and I'll turn things over to you, Albert.
All right, thank you. And let me first start by saying thank you to Amy and Nigel and Kenzub and the other people with the Plant Gene for making this happen. So this protocol is specifically for rice, the cultivar Nipponbare. And so this is perhaps one of the easiest protocols for a cereal crop that I know of. So if your research is directed towards a cereal crop, and rice would suffice for that, this is an excellent protocol for generating events. So one of the advantages of this protocol in comparison to many of the other monocot or cereal crop plants is that it can be initiated from mature seeds. So you don't need to be growing a stock supply of immature embryos that you need to maintain, but rather you can start Callus directly from mature seeds anytime you need these for experiments. And this produces a very early embryogenic Callus, and I can talk more about what I mean by that in terms of early later. So yes, you can get your seeds from Dale Bumpers if you need that, and there is the link to that site for the Nipponbare seeds. And one of the reasons for doing this cultivar in the first place is that it was one of the earliest genomes to have a high quality sequence available. And this has been the foundation for many of the other cereal crop genomes that have been published. So Nipponbare is the reference genome for rice. Okay. So starting from mature seeds, and this is really great, you can store these under proper seed storage conditions and they'll last for quite a long time. So these are manually dehusked using fine grit sandpaper that you can just buy at a normal hardware store. But it is the fine grit, I must say. And this is done, yeah, you can go to the next one. So this is done by just lightly sanding the seeds with a separate piece of paper and then picking out the seeds that have been dehusked but are not otherwise harmed. So you want to pick out non-broken ones, and these would be surface sterilized in 50% bleach, usually adding a couple of drops of tween. I think, did we miss a slide, Amy? Or was that on the last slide? About, no, back one, I think. Yeah, so sterilize them at 50% bleach for 30 minutes on a shaking rotary 200 RPMs at 28 degrees, and usually a couple of drops of tween as a wetting agent. Okay. And then these surface sterilized seeds would be plated on the induction media and just space them out, and then seal the plates with parafilm and incubate at 28 degrees in the dark. And this will cause the seeds to germinate. Next slide, I think. Oh, so this is the Callus induction media. Let me just a few remarks about this. This is using N6 salts. For those of you that aren't familiar, that was a medium that was developed in 1975 by Chu, and you can look up the constituents for this, but just some hallmarks about it in comparison to MS is that it has no ammonium nitrate and has rather in place ammonium sulfate. And so this media will induce a very early embryogenic culture. There's some other distinguishing factors about this, but this is your Callus induction media.
So we didn't have any questions about the components so far. So let's go through the callus induction
Yeah.
section, and then we'll take questions before we move on to talk about infection.
I actually could also, if you go back one more, Amy, back to that. Yeah, it also has proline in it, L-proline, which also, I think this media regime in general is encouraging early embryogenic callus growth in rice. The medium itself was designed and used for anther culture and microspore culture for generating haploid callus. So for this function, though, it works really well at this stage. I don't think there's anything else I'd like to remark. Oh, except for this is also using N6 vitamins and not MS. Unless anybody has any questions on that, we can go on. So the seeds will eventually germinate, and you should go in and remove any of the germinated roots or shoots and allow the callus to grow for another 2 weeks or so. And it'll form a nice early embryogenic callus after about 2 weeks. And those are, so those can be just removed and then cultured separately and subcultured every 2 weeks and expanded. So eventually you can expand out this callus, subculturing it to keep it healthy about every 2 weeks. And so you can maintain a stock of embryogenic callus that then can be subcultured before it's used in transformation. Next slide. Yeah, somebody just commented about proline. You can look that up in terms of an amino acid addenda to media. Generally, it's used to reduce stress response. And I think this was the intent on its original addendum in the anther-culture media. So we keep that in this media throughout callus induction and beyond. Remove the roots, remove the root of germinating seeds. Yeah, you want to remove both the root and the shoot of those germinating seeds, as they would contribute to disrupting the hormonal response that you want to encourage by this media, which this is at 2 mgs per liter of 2,4-D. Yeah, proline is also an osmoprotectant. Somebody pointed that out. Oh, but what you will generate here is a stock culture of embryogenic callus that'll look about like this. And you should use a dissecting scope when subculturing this to always subculture the healthiest embryogenic callus. You'll see other cell types that should be avoided when you're doing this subculturing. So you can maintain a stock culture of these for 3 to 4 months. If you're really worried about somal clonal variation, you might want to start Callus just initially and then transform it directly. But for most cases, for most ways that you would use this, for 3 or 4 months, you will not be disrupting regeneration capabilities at all by doing it this way. So before infection, then, starting with your stock of callus, you can divide these into 1 to 2 millimeter pieces of embryogenic callus, put about 50 of them, 50 pieces per plate the day before infection. You would transfer these small pieces to media that contains the acetosyringone overnight to make sure that that is saturating the culture. Somebody's asking a question about other types of rice, japonica, etc. I could talk about that later. This protocol is going to work really well for Nipponbare, and we've investigated other genotypes as well. And I think I will stand by this protocol for Nipponbare, and as far as how it extends to other indica or japonica varieties, you would just have to check it out to see. But I think you will have relatively spotty results. And I didn't want to bring this up until later, but I think in combination with morphogenic regulators, particularly GRF-GIF, this protocol will work well for other cultivars as well. So going on, though, with the infection media. So this is also with N6 salts. The increased sucrose and glucose there is mainly to keep the agrobacterium happy. Notice that this is on a slightly lower pH and it's not been autoclaved but filter sterilized. Okay.
Thanks. So before we move on to talk about the co-cultivation, there are some questions in the Q&A box and in the chat. So just a reminder, if people could use the Q&A function, it's at the bottom of your screen. That just helps us see what questions we haven't answered yet, and it keeps things a little bit more organized than asking your questions in the chat.
Yeah.
So I'll start off with this question from Joshua Clem. Is acid casein peptone a suitable substitute for casamino acids?
I haven't tested it. So presumably that would be the case. But I think I would stand by this protocol, and anything you want to test is up to you. But get back to me to see if how that works out. Presumably that should be fine, though. Yeah.
And then another question about casamino acids from Joanne Connor. Are the casamino acids added after autoclaving?
No. No, that's added before autoclaving.
Okay. And this one is from Nicholas Figaro. Is there a difference between using agar and gel-right?
Yes. Gel-right for this protocol, I'd stand by that too. Phytogel might be a decent substitute, But if you can get gel right, I would go with that at 4 grams until
Okay,
you get
thanks.
to the last regeneration step. But yeah.
So this question is from Lauren Powell's. Do you move the small pieces individually, or can you use a filter to move them together?
We move them individually. So um, you can test that. I think when we did that, it lowers your transformation frequency, but it's easier to move them. I think you'll find that this is not a hard subculture, that you're moving 50 pieces, but it's going to go very rapidly.
So this is
And I
another
think,
question.
to add on to that, I think you'll have less of a problem with Agrobacterium overgrowth by moving them directly onto media. But yeah,
Instead of using a filter.
yeah.
Have you ever just taken the petri dish and kind of dumped the cali out?
Yeah.
Okay.
You know what though? We've always wanted to pay attention to transformation frequency per piece.
Okay.
So that's why we didn't mix everything up like that. For ease of doing this, if you don't care about that number, then yeah, I think you could group them all together.
Okay.
Yeah.
This is a question from Mr. Rue. How do you determine the piece of callus, size of callus, and infection volume?
Um, well, as I indicated, you're taking pieces that are about 1 to 2 millimeters across. So these are fairly small pieces of callus, and you've maintained these by visual selection for uh, their embryogenic quality. So you know that these are good pieces of callus. And the volume that you're spotting with the agrobacterium is empirical. We do that so that it's a volume that you would see by adding it with the pipette, that's about 1 drop per piece of callus, and that's all that you need. You're not flooding the entire plate with agro, or else it'll overgrow.
Thanks. This is from Fazio. Sorry if I mispronounce anyone's names, by the way. Yeah, apologies for that in advance. So their question is, why is the pH of infection media 5.2, which is lower and not 5.8 like other media types?
That's that was to promote the health of the agrobacterium. So arguably you could do that at 5.8 and keep your cells happier, but it's a relatively short exposure. So you'll see other protocols where the infection is at 5.8 for agro, but we felt in this protocol for EHA 101 and 104 that this was better.
Great, thanks. There are some other questions. Some of the questions pertain to things we're going to be talking about coming up next, so I think we'll answer those shortly in the presentation. But if not, I'll come back to these at the end. So let's go ahead and move on and talk about the infection media.
Yeah. So this was using EHA 101. You can try other agrobacterium vectors. And sorry that this was blocked out, but what we normally do is grow the culture and then have blank infection media to dilute it down to the appropriate OD. So by appropriate OD for EHA 101, we were using 0.3 to 0.45, which is slightly lower than what you might want to use for, say, AGL1. But you can dilute it down to this using your blank infection media. Yeah, so co-cultivation then, also in N6 salts, N6 vitamins with the proline, back to 30 grams of sucrose, including the 10 grams of glucose, favoring still agrobacterium growth. So, and this is again on gel rate.
Okay, so sorry, before there were a couple of questions about infections, so I'm just going to go through those,
Yeah.
and it was a short section,
Yeah.
before we move on to co-cultivation. So, how long of infection for Agrobacterium a solution on the cowlie? What is the best time to prevent overgrowth?
Well, that is a good question. Um, we have varied that exposure from an overnight exposure to 3 days. So there's a wide range that you could use. 3 days is overdoing it. Um, I think in I think a good rule of thumb would be 24 hours at 28 degrees
Thank you.
with this OD.
Okay, yes, that's an important part.
Yeah. If you lower the OD, you can go longer, but yeah, I stick with this.
So this is from Wakar Ali. Where did you store your prepared petri dishes, including media before transformation, at 4 degrees or at room temperature?
The you mean media without anything on it? Is that what you mean?
Yes,
Or
yes.
we
Yeah,
store
so you
ours
make
at,
your media
yeah,
ahead of time.
yeah, we
That should
store
be
ours at
stored.
4 degrees.
Okay, great. Okay, there's a question about co-cultivation. We'll be talking about that shortly. And so we had a question from Abida about using GV3-101, and we also had some questions in the Q&A about using AGL1 and EHA105.
Yeah,
So yeah, if you want to talk about those a bit, I do just want to stress that, so we're going over this protocol and kind of like Albert alluded to at the beginning, we're standing by the details in this protocol. So if you're making any changes, then you're doing so at your own risk. But would you like to talk about using other agrobacterium strains other than EHA 101?
yeah. Now this is fine, and you can experiment with this, knowing that rates of growth of these are going to change, and that will affect your infection time or your cultivation times, so that aggro won't overgrow. If you see the aggro starting to grow visibly on your culture, that's too late. So you can do this sort of empirically by, if you're say using AGL1 and an optical density of say around 500, you would want to pay attention to how quickly that agrobacterium grows. Um, Also, different agrobacterium strains will affect um, copy number for insertion. Um, some people report, say, higher copy number insertion rates with AGL1 in comparison to EHA 101 or 105. So paying attention to those parameters. Another thing would be that various vectors themselves can affect agrobacterium growth. Uh, we've seen this, for example, if your tDNA is carrying an especially large payload, that can affect growth of the aggro. But as Amy suggested, we're standing by this one. So I think you would be successful with really any of those other agrobacterium strains, as long as you're paying attention to, selection, what are you going to use to kill your agro. In this protocol, we're using carbenicillin and timentin, but you can use other selective agents for your agro as well. Hope that helps.
Okay, great. So we are going to move on and talk about co-cultivation.
Yeah, so once you have these plates prepared, it's fairly easy with, we've even used repeater pipetters to dispense about, 5 microliters per piece of callus. You don't want your agro to spread out over your media, so about a 5 microliter drop is sufficient. And then we just let that air dry in the hood, so that you're not swimming in agro at that point. And other protocols use air drying, even on immature embryos, as this seems to help the agrobacterium adhere. So this one was using 3 days co-cultivation, and then but you might want to reduce that time depending on your OD of your agro. But then that's transferred to the resting media with the antibiotics to kill the agrobacterium. And so they're going to remain on the resting media for 7 days, and you don't want to disrupt the tissue at this point. We are paying attention here to transformation frequency per piece. If that's not an issue for you, then you don't have to be as careful about that. So your resting media then is still on your N6 with 30 grams of sucrose, with 2,4-D at 2 mgs per liter, and N6 vitamins. And now with your carbenicillin timentin. And that would be for a week.
So there's a question from Rebecca Torsky. Do the cow not need a wash step before resting media step?
No, we didn't find that this was necessary. Matter of fact, I think I'd avoid it.
Okay, why is that?
You wouldn't want to disrupt the tissue. It's easier just to move it.
Okay.
I think if you need to wash it, you probably would do better by reducing your agro concentration or modifying the time that you're spending in co-cultivation.
Okay, thanks for that clarification. So this is a question from Jack Brooks. So this is just not necessarily about co-cultivation, but what is the normal transformation frequency for this protocol?
Yeah, per piece of callus, we were experiencing always upwards of 20, 30%. So this might vary on your stock of material. So you're getting plenty of transformants per treatment.
Okay, great. And then this is a question from Carlos Montoya about agrobacterium growth. So how long can agrobacterium persist in tissue culture after the infection? Do we need to autoclave the soil used for growing plants after we are done with the experiment?
Um, Once you've transferred material into soil, we've not monitored that. But in fact, we can see agro still growing on plant material after regeneration 3. So we have tended to keep carbenicillin and timentin through to the second regeneration phase because of this. So with some agro strains, it can persist in the media for uh, quite some time, which practically speaking, this also is the reason why you can't PCR off of Callus directly, is that you can have residual agro there that would um, contaminate your PCR results. So we
It
don't,
may,
yeah,
I might say that question about autoclaving soil, that's going to depend on the regulations or
that's correct.
within your institutes. So be
Yeah,
aware of.
thank you for that, Nigel. Yeah, sorry, was there anything else you wanted to add about that? I just went off video, so I think no. But yeah, like Nigel said, so that's something you need to consult with the biosafety group at your institute. Okay,
Yes,
so
biohazardous waste, correct, right.
yeah, I always autoclaved it just because I don't know, trying to separate out the plant material from your soil is was, it's easier to just dump everything.
Yeah.
But yeah, again, check with your institute. Okay, so with that, let's move on and talk about the resting phase. Or sorry, I think we're at selection.
Yeah, so we have used both hygromycin and PPT. So depending on your vector, of course, you'd have to have these selectable marker genes there. But hygromycin is very clean in terms of selection for HPT expression. So 50 milligrams per liter of hygromycin in selection one is adequate for this. Um, and we use the bar gene because of its added advantage of doing the paint assay on regenerating plants that you can apply by allophos to leaves. But for depending on what you want to do, they both work well. So in selection one, you're either applying 10 milligrams of PPT, and if you're in bialaphos, that would be 3 milligrams of bialaphos. Or if you're selecting for HPT, then it's 50 milligrams of hygromycin. And that's an either or here. I want to make that distinction. It depends on your vector. Um, if you use both, you'll kill both. So just to be clear.
Things that yeah brings me to a question from the chat from Alabakash. So using double antibiotics, can it suppress regeneration ability? Have you tried using a single one and
Yeah,
seen
yeah.
its effect?
No, wait. Only use a single one.
Okay.
If you have both selectable markers in your vector, don't do that. I hope this wasn't confusing. This is an either or situation depending on your vector.
Okay, great. Thanks for that clarification. This
Yeah,
question was asked a few minutes ago, so I think that was
yeah,
before, you made that comment about it is either or. And
yeah,
you'll see that in red there at the bottom. So just one.
yeah.
And this is another question from Joshua Clem. Any concern about 2,4-D crystallization with storing media at 4C? I've noticed the liquid stock crystallizes at low temperatures.
If you have obviously if it's crystallized in your refrigerator, you want to bring that to temperature before you use that as a stock solution. But we have not seen a problem with it crashing out of our media at these lower concentrations.
Okay, great. Thank you. So Anthony Kigaru has a question. Why is hygromycin concentration constant? I thought it should gradually increase.
Whoa, that's a good question. The reason why you can get away with that in Nipponbare rice is because I alluded to it's a very early embryogenic culture. And what this explains is that you can literally kill everything around almost a single cell will recover uh, using this protocol. So hygromycin is a very quick killing agent. And um, if if you tried this on more organized cultures, like maize, for example, or sorghum, you would have a lower recovery of transformants. So the fact that you're using a high level of hygromycin here means that you're able to kill almost everything around an event very early on and recover a stable colony from that. That's a good question. Thanks. I hope that's an adequate explanation, but whereas PPT is going to kill very much more slowly, you know. So yeah, that's an explanation. Thanks.
Great. Yeah, if you have a follow-up to that question, feel free to yeah, put right again in the Q&A. This question is from Abita. How important is it to maintain temperature at each stage?
Oh, absolutely important. I would say you want it, you don't want this to vary much beyond 28 degrees. Um, for some protocols now, especially if you're going to venture into using baby boom and mushel, you might be subjecting Callus to heat shock. I've not done this with Nipponbare, so I don't know about its recovery. But owing to my explanation before, we didn't experiment too much above or below 28 degrees. So I would stand by that.
Okay, thanks. And then there have been a couple of questions about concentrations of the different media components. So I just want to clarify that this is per liter, correct?
Right,
Okay.
correct.
Yeah,
Yep,
so that should give you your answer. So this is so someone had a question about I've seen a carbonicillin.
yep,
So that would be 200 milligrams
per
per
liter.
liter.
Right.
Okay, great. Um, and then this is a question from Ashkan. You showed that you added acetosyringe to the medium. Did you also add it to the agrobacterium culture?
Um, it's in the infection media. Is that what you mean?
Um, I think so,
So
is what
yeah, it
they're,
would be in
yeah,
the
so,
infection medium and in the co-cultivation medium. So when the agrobacterium sees the acetosyringone in the infection medium, it's becoming activated. Yes. So you'd make up your infection medium with the acetosyringone added.
okay. So Alabakash wrote back with clarification on his question. So my question of using two antibiotics, parentheses, carbenicillin and timentin, was during resting phase. Use of two antibiotics to suppress agrobacterium can also affect regeneration ability sometimes.
Yeah, that's
Have
right.
you tried using a single antibiotic like carbenicillin or timentin alone and see effects on regeneration?
Yeah, in fact, carbenicillin alone will work. We included timentin here because it improves regeneration.
Okay, that's interesting.
Yeah.
Okay, there you go. So, yeah, yeah, good question about the Timentin. Okay, great. So, sorry, I think I had the slides out because I had a slide about selection one, and then we're here at selection one. So apologies if that was confusing to anyone. But let's go ahead and move on and talk about selection one.
Yeah, so you can see from this slide that hygromycin on the right here um, produces a really nice embryogenic callus that in our hands, each one of these is one event. So we very rarely get a mosaic of events, even though this and so this is why also we use a small amount of callus to start with. So generally speaking, one of these colonies is one transformed event. So when this is a very interesting distinction. Yes, when we move to regeneration 1, we're transferring this from N6 salts to MS salts. And so I've already alluded to the fact that the main difference here is that MS medium has a higher level of ammonium nitrate. Now, when you look in the literature about this, we would see that this transition from N6 to MS favors development and organization in general. So I am not sure that the physiological response has been clearly defined for that response, but you'd see this over and over again, that MS salts tends to encourage organization and later stages of embryogenesis. So you would recognize that right away when you make this transfer. We're also dropping out the L-proline, and sorbitol is now included as a carbon source. This also encourages development and later stages of embryo development and the beginnings of shoot organization. So this transition from N6 to MS salts accompanies and encourages later stages of development. You're also dropping out the 2,4-D, which will encourage shoot development, and you're increasing, you're including now kinetin and a small amount of NAA. Those are all favoring shoot regeneration. And you're keeping in the selective agents. So you would recognize shoot regeneration pretty much right away in the first week after transferring them to a low light of 16 hours. In the first 2 weeks, you'll start to see the greening of the later formed embryos and the beginning of shoot regeneration. Once the shoots are well formed, they can be then transferred into a plant con with regeneration medium 2, which will favor root formation. And again, this is a fairly robust protocol, so you should be getting at this point a fair amount of shoots to transfer. So in regeneration 2, if you're using PPT, we keep that in the media, as we have encountered escapes with by dropping out the PPT, and we don't see that keeping it in reduces frequency. In other words, you might think that you're helping them out by dropping out that selective agent. You're not. If they're transformed, they will survive this quite fine. We drop out the hygromycin because it's expensive. And by that point, hygromycin is such a clean selective agent. Now remember, this is an either or. You're not doing this at the same time. If you're using PPT as your selective agent, keep it in. If you're using hygromycin, you can drop that out in regeneration, because whatever is survived at that point is transformed, and we don't see very many escapes. So that's the reason for that. Oh, and you're keeping in the MS salts and the MS vitamins. In case you're not familiar with the difference there, N6 has no myo-inositol, and myo-inositol will favor shoot regeneration. Also, N6 has no thiamine. Or I mean, N6 is higher in thiamine than is MS. And so dropping that out is also favoring shoot regeneration, we think. Okay, those are my comments on N6 versus MS.
Thanks. So we have a couple of questions here. So first, from Ritesh Kumar, any comments or suggestions on trying trans-zeatin in the regeneration media?
As opposed to kinetin. I think that'd be worth looking at. We have not done that. Yeah, I think I'm not sure if that would favor this or not, but that would be worth looking at. We've not looked at it.
Okay, thanks. I have a question from Lucas. In the regeneration 1 stage, do you mean low light? Does that mean near 20 micromole per meter squared? Or meter squared per second.
As opposed to 50 or 60 later on, yes.
Okay. And then a question about, this is from King Shen. You use N6 salts and N6 vitamins. Do you prepare these as stocks or use commercial? If the latter, could we just purchase N6 media, which contains N6 salts and vitamins?
Yeah, Phytotech might sell these separately. In the old days, we made our own. But, if you can buy them separately like that, buy them. Great. Yeah.
All right. So this is a question from Mr. Have you checked the putative transformants using gene-specific primers? I guess that's a question about how you confirm your transgenic plants.
Well, yeah, again, this is a problem with agro-contamination, right?
Mhm.
So, if that would be your major problem with doing that. Um, you might be able to figure out a technique where you could PCR off of an integration site to determine it like that. But why not just wait a couple of weeks? And then you'd have you know, by the time you get a transformed shoot that's large enough to do direct PCR, um, you're probably not losing that much time. I don't know what your study is involving, but you'll generate plenty of shoots here in order to do an assay like that fairly early if you need one.
Okay, great. Another question from Alabakash. How often do you come across chimeras using this particular protocol?
Yeah, relatively very rarely. And I know this because at one point we did a colorimetric study doing this. So if you wanted to make those, I think it'd be very difficult. But this is also why we start with very small pieces of tissue. We want each event to be its own event without chimeras just complicate issues. So, yeah. So it's relatively rare.
Okay, and then do you have any suggestions for, this is a question from Abida. What could be the possible reason that I'm not getting any shoots from the regeneration stage? Calli are not dying, but not growing or greening as well.
Oh, this could be a problem with your embryogenic callus that you're starting with.
Mm,
If you're generating embryogenic callus, you might want to, at that phase, just take some and take it through the regeneration to check it. If if you're subculturing non-regenerable callus from your original callus induction from your mature seeds, if that's not regenerable at that phase, you're not going to get it back. And it won't, the media will not recover non-regenerable callus. So, and you can get non-regenerable callus from the roots, by the way. Root tissue left on in that early stage will generate callus that will grow, but it will never regenerate. And it'll look different too. You'll see this as more or less translucent cells. Oh gosh, I don't know how else to describe it. They'll look kind of crystalline, and they'll grow rapidly. You can see this in other monocot cultures too. But I would suggest that probably you do not have embryogenic callus that you're taking into the situation from the beginning. That'd be my guess.
Thanks. Yeah, thanks for that clarification. Yeah, the health is really important of your starting material.
Yes. Regular subcultures of that is essential. 10 to 14 days, 14 days being at the outlier. Right.
So, this is a question. I think you might talk about this in regeneration too, but this is from Wakar Ali. Can we mass multiply the transformed embryos at the early plant stage using basic tissue culture technique?
Yeah, so if you want to expand an event and get many more T zeros is what I think the question is. Sure, go for it. You can take a transformed colony and in a month or two expand that. I'm not sure why you want to do that, but yeah, you can generate any number of clones from one of these colonies by expanding it. And you would want to keep your selective agent in that media as you're expanding it.
Okay, thanks. Yeah, I had a following up with that. So, because I think I see this in CRISPR papers sometimes where people split the calli
Yeah,
and then, they happen to get, a biallelic editing, and then they have 5 plants from the same event.
right.
But now it's 100% editing in these 5 plants.
Oh,
But
right,
I think it's
right,
not.
right.
Yeah, I think just my experience, you have to be careful with splitting the plants because you're kind of giving yourself more of that
Yeah,
event
yeah.
that might not
And
be
it's just
accurate.
really one event expanded over 5 times or 100 times. Yeah.
Right,
And you don't want to inflate your efficiency numbers.
right.
Right.
And then this is a question from Ritesh. Do you think desiccation can help improve the regeneration process overall? Also, do you have any comments on using phytogel instead of gelrite to enhance regeneration?
That question came up earlier about gel right. I don't know. Um, you notice that in this protocol, we lower the concentration in regeneration too. That's mainly to promote root growth, but I would I would just guess that that substitution would work, but you'd have to try it and get back to me.
Okay, yeah, thanks. And then the other question was, do you think desiccation can help improve the regeneration process?
Desiccation, we do the air drying when in the early phases of agro infection. I don't know if desiccation during regeneration 1 or 2 helps you for that phase of regeneration. For Nipponbare, though, this is not a problem. If you're experimenting with um, other varieties, then yeah, I think this might help you. Um, I also know from one of my colleagues that this protocol works really well with GRF gift vectors on indica and some other recalcitrant varieties, where you would get a proliferation of shoots using a GRF gift-like vector. I know that's a little bit outside of this workshop, but if you are investigating recalcitrant rice varieties, and so if you're looking at desiccation to encourage regeneration at this phase, that might be another way to look at this. It would be to improve your vector in the first place, maybe by looking at GRF GIF. I don't know if that helps, but
Yeah, that answers the question. And so someone had a follow-up question for which desiccation agents can be used in regeneration media. Are you using a desiccation agent or are you just letting it dry?
um, letting it air dry is what we do just on filter paper, and it looks kind of brutal. But if you're going to do this, say, again, I'm talking about morphogenic regulators here, but for if you're using baby boom and mushel, for example, and you want to excise that out, some people have used desiccation sensitive promoters in order to do that. Um, so uh, we know that desiccation can be used for either removal of baby boom and mushel, but also to encourage regeneration at that phase. But that's just an air drying technique. Um, you could include, some people have included osmoticums in order to do this. But again, that would be up to you to experiment with.
Great. All right, so let's move ahead with regeneration 2.
Yeah, so once your shoots are regenerating in plant cons, we harden them off by transferring them to 4-inch pots in soil in a walk-in growth chamber, and then hardening them off by removing the plant con lid for several hours at a time per day until you regrow a cuticle. And then these are grown back in 8-inch pots and then watered from below in trays. We can do this with rice adequately, and it lowers the greenhouse maintenance. Um, it does somewhat encourage fungus gnats and aphids, and so you'd want to pay attention to your greenhouse to make sure that you control that. But watering from below in this case is great, and we use Metromix 510 for doing this. So plants then are grown in the greenhouse, generally with 16-hour day length, and watering from below. So we grew amazing numbers of events like this that you see here. We were, for this study, making a transposon mutagenesis library, and we're interested to grow as many events as possible. When you do this, though, I would admonish you to pay attention to your labeling. So this is more of an issue with keeping your event straight than anything else. And so, but anyway, it's a very robust protocol for producing large numbers of events if you need them for studies like this. And those are your T0 transgenic events, selfed, and you can then harvest and save your seeds adequately for a long period of time.
So we are at the end of the presentation and then right at the end of the hour. So we are just going to wrap up with a few more questions. The first one is from Lawrence. Did you lose many plantlets during acclimation to soil?
In fact, that's a good question. Generally, no. I would have to say if you're paying attention to there are some critical factors though. When you have to soak off the agar, you don't want to just simply plant them. The agar has um, a carbon source and your fungi will grow. So you want to take the plants out of the plant con into a tray of water and then gently tease away all of the agar so that you're transplanting that without any agar on those roots at all. And you don't want to spend too much time with those being exposed to the air at that point. They have no cuticle and they will desiccate very quickly. So I think if you're paying attention to those details in comparison to any other transgenic plant, no, it shouldn't be any different.
Great. And then I think this question, we might use one of your question slides for this. How do you distinguish between embryonic callus versus a regular callus?
Oh, yes.
Yeah.
That,
So we were
you
just
know,
talking about this.
I did.
So this is
Oh,
the
this is a
extra
great slide
slide
for
that
this.
Albert's
Now,
prepared.
unless you're used to looking at embryogenic callus a lot, these distinctions are um, not clear to someone who might be new to this. So when you look at this callus, you see that it's comprised of a lot of BB-like shaped very small clusters of cells. One thing that you don't see here is a defined epidermis. You don't see large structures that look like they're ready to become shoots. If you're familiar with maize or wheat transformation, you would see what we would call type 2 cultures developing into more organized type 1 like cultures. Type 1 cultures have more organized tissue. The reason why this is significant is that if you have organized tissue there already, um, with say a mature epidermis, an epidermis you can see from basically under a dissecting scope. Imagine that you had transformed an epidermal cell. When you apply selection, that cell is going to die along with the other epidermal cells. It cannot sustain itself. Early embryogenic cultures have this property of self-renewal without depending on the rest of the culture for their survival. I don't know if that's an adequate explanation. Maybe give me the next slide, because I did
Okay.
this figure for a publication, and maybe this will help to illuminate this. These are the stages in row A of zygotic embryogenesis in maize. The second one is on somatic embryogenesis, also in maize, as initiated from epidermal cells on a immature embryo. Um, I don't know if this adequately describes the situation, but you're going through the same developmental sequence as in a zygote. And in maize, terminal differentiation occurs in the embryo at day 6. That's the third stage over. And this relates to selectability of early embryogenic cultures. And I could take a whole hour to explain this, but I hope that's adequate. It's a pet subject of mine for sure. But
Great. Well, that was a great question. And yeah,
yeah,
thank you for asking. Glad we
yeah,
got to cover that.
yeah.
Okay. So here is a question from Carlos Montoya. How do you test the lines for homozygosity? Is selfing enough?
Um, well, selfing in rice is so predominant that you would expect that to occur. So, and actually outcrossing would be harder to actually perform. But you would definitely want to check this. Um, so um, you could do that by a variety of methods. It depends on really uh, what your needs are for the study, I think.
Great. All right,
If you're
thanks.
doing that for editing, definitely I would do that by sequencing. Uh, make gDNA out of your early events and have that sequenced through your target.
All right, thanks for that advice. And then this is going to be our last question because we're just a bit over the hour. So this is from Ken Wan. Do you see albino plants?
Oh, Gang Wang. Yeah, of course. We do see albino plants. And can you ask her what the reason is for that? Um, we this is a low frequency situation. And we can see this I can tell you it doesn't matter about the selectable marker. We can see this with PPT. We might see it more with hygromycin. And we see this with other monocots too, with various frequencies. Switchgrass, turf grasses come to mind. We can make a lot of albino plants. Not a lot, but yeah, you do see this. Yeah.
You do see that. Okay.
Yeah.
And then Joshua commented, I've noticed this in plants that shouldn't be albino, perhaps mutations resulting from tissue culture.
Um, yeah, I suppose. I'm not sure this has been, I don't know the literature on this, of whether this has been clearly defined
Mhm.
reason for this, or even what the mutations are that result in that. I've
Okay.
speculated, but
It's
no.
unclear. Yeah.
Yeah.
All right. Thanks. All right, so that is all. Just want to thank you everyone for attending today and contributing. We are going to do a quick poll. So just let me pull that up. So you should see this on your screen. So we want to know if you are interested in a follow-up session to today's presentation. It's not a guarantee that we're going to do a follow-up session, but we are thinking about doing another rice transformation event. So please take a moment to give us your input. That's really important to help the steering committee plan these events. And then a reminder, if you are a planting member, you can access the discussion forum. If you go to the website and log in, it's one of the first things you'll see on the page. All the information is there on the website and how you can participate, and you can ask a question. Your email goes out to the planting community. So it's a great way to connect and carry on the discussion today. If you had a question that you weren't able to get answered today, we can continue to the discussion online in the forum. And I just want to say thank you again for joining today, and I look forward to seeing you guys at our next event.
Thanks, everyone.
All right. Thank you again, Albert. Really
Thank you.
appreciate you sharing your expertise with everyone today.
Thanks, everyone.
Lots of little hearts coming up at the screen, if you can
Right,
see those.
right. Yeah. Thank you, everyone.
All right.
Thank
Thank
you,
you,
Albert.
everyone.
Right.
Bye.
Thanks,
Bye.
everybody. Bye-bye.
PlantGene would like to thank the National Science Foundation for their support. To learn more, please visit their website at nsf.gov.